Hi, my name is Ravi Vij. I'm from My Cancer Haven, and today in our Conversation with Cancer Experts series, we have the pleasure of having Dr. Larry Anderson, who's professor of medicine and directs the myeloma, Waldenström's, and amyloidosis effort at the Harold C. Simmons Comprehensive Cancer Center at UT Southwestern in Dallas. We're fortunate to have him, and he's going to talk to us about a CAR T that he has worked on, which has gone through different names, but currently AZD0120. It's a FasT-CAR. So, Larry, welcome.
Thank you so much, Dr. Vij. Glad to be here and have talk about these exciting new products.
So let's get started. So I think this product was originally with a company called Gracil, and so there was actually two data sets that were presented at the American Society Hematology meeting in 2025. One from the Gracil early experience, and then the compound has now been moved into co-development with AstraZeneca, and there was another presentation. So we'll go through both of these presentations. But first, starting out with the original, presentation by the Chinese group, can you first lay the background to tell us what this car is, how it is different, and what a FasT-CAR means?
Yeah, so there are two major differences between this product and the other products on the market for CAR T for myeloma. One is that this uses a rapid production, rapid manufacturing protocol so that these cells are essentially manufactured in just a few days and they can be available for the patient after rigorous testing within about 2 weeks instead of the usual 4 to 6 weeks. And also, these particular CAR T cells from AstraZeneca, the AZD0120, have two separate targets. So they're dual targeted CAR T, both BCMA that's on plasma cells and CD19 that we normally think of as a B cell target. So most B cell malignancy CAR Ts are directed at CD19, but also the putative myeloma stem cells also may express some CD19. And we're hoping that this will help prevent that reemergence of myeloma over time from myeloma stem cells as well.
So one of the things also about FasT-CAR T, obviously everybody talks about the fast as it's fast to get to the patient, but there's other, I think, benefits to it. I'm told that it actually, with this procedure, you actually get some of the immature memory cells that may actually be more important in increasing efficacy as well. Though we don't have proof of that, can you elaborate on that?
Right. I think one of the problems with, manufacturing that takes many weeks is many of these T cells, by the time they go into the patient, are exhausted, no longer fit. And if we have T cells that are in the process of being expanded and early on in the process, these T cells hopefully will be much fitter, much more different phenotype that's able to generate memory T cells and potentially live longer within the patient, hopefully.
So now just delving into the presentation by the group from China, what can you tell us about the characteristics of the patients that they studied?
Yeah, and in this particular study in China, there's been a couple different studies reported on this product from China. This one was in a newly diagnosed population. So this is for patients that were a mixture of transplant eligible and transplant ineligible frontline therapy. So most of these patients had not received prior therapy. About 10% of them did have tumors or plasmacytomas. They also tested the apheresis at a few different time points. Some of the patients had their T cell collections before their induction therapy, some after one cycle of induction, some after two cycles.
So how did it look in comparison to others in terms of safety?
Yeah, so the safety profile looks excellent. We're really encouraged by this. A fair amount of the patients, about 30%, had grade 1 cytokine release syndrome, but only 3% had grade 2, and no patients had grade 3 cytokine release syndrome. Also, most importantly, essentially no neurotoxicity, and especially no delayed neurotoxicity like Parkinsonism disease, et cetera. So that's quite exciting.
So, and what are the preliminary results?
Well, it's sort of hard to beat 100% overall response rates and 97% stringent complete response rates. So really, and maybe even the few percent that weren't already in a complete response, it's still early, could potentially even convert as well. So that's pretty impressive.
And then one of the hot things is MRD these days, measurable residual disease. So how did this fare there?
Right, so looks great there as well. So out of those that were evaluable, so not every patient has a baseline clone identification. Out of those that had samples that were evaluable, 30 out of 30 or 100% were able to achieve MRD negativity at all dose levels, at least one time point. And then 25 out of 30 or 83% had at least 12 months of sustained MRD negativity as well.
And what do we know about the durability?
So far, with the 36.5-month follow-up, the majority, over 89%, were still in remission and alive, and that's also very impressive as well.
Yeah, certainly looks very impressive. And I guess one of the things we know is that MRD negativity perhaps at one time point may not be fully indicative of how a patient is going to do long term, and it's sustained MRD negativity that perhaps is more informative. So, what do we learn from there?
Yeah, correct. As these graphs show, it really matters more if you have sustained or, multiple time points of MRD negativity, not just one. And some would argue that if it's just one time point and you lose it quickly, that may be worse than having no MRD negativity at all.
So let's move on to a trial that you're more familiar with having participated in it. And this is the new incarnation of the same molecule, AZD0120, as it now is being developed by AstraZeneca. And this is, a trial looking at the same CAR-T in relapsed refractory myeloma, and it's a phase 1b/2 study. So, what can you tell us about this trial design?
Yeah, so this design is for patients who've had at least three prior lines of therapy, triple class exposed, PI, IMiD, and a CD38, so not for the frontline setting in this trial. Um, must have had progressive disease. So this is essentially, even though this is only 3+ prior lines, this is these days, some heavily refractory patients fit into this category now because we're using so many different classes up front in the first two lines.
And what were the demographics of the patients that were accrued to this study?
Yeah, so these are heavily pretreated, really high incidence of high-risk cytogenetics, 35%. Um, a small amount of extramedullary plasmacytomas, not a huge amount, I think 4%. The majority of these patients had received a prior autologous stem cell transplant. The majority were triple class refractory, and only about 27% required bridging therapy because of that rapid manufacturing turnaround in the study.
So in this experience, how was the side effects that we always are looking out for in CAR T cells, the CRS and the neurological toxicity?
Yeah, so again, similar to the experience with the other study, we do see mostly grade 1 cytokine release syndrome, but very low incidence of higher grade uh, cytokine release syndrome, a little bit of grade 2, no grade 3. And in this study, there was one grade 1 ICANS or neurotoxicity event. But again, I would say the major finding is that we don't see any of the delayed neurotoxicities, no Parkinsonism, facial nerve palsies, Guillain-Barré, no IEC-associated colitis. So none of the things that most patients are really worried about, and mostly just easily manageable low-grade cytokine release syndrome.
And how did it fare in terms of effectiveness?
So again, in this heavily refractory, heavily treated, heavily refractory population, 96% response rates, 78% complete response rates in the setting of excellent safety. So that is also very impressive. And if you look even at the subgroup of patients who had been exposed to previous BCMA-directed CAR T cells, we're still seeing 100% response rates and 80% complete response rates.
And once again, the MRD negativity rates appear to be pretty high.
Yeah, so the majority, 94% that were evaluable, were able to achieve MRD negativity, and most of this was within a month, so very rapid onset.
So I understand going forth with this CAR T as well, there's a very large program that is currently in uh, process of rolling out that there are even in the United States plans to move this into the very front line. Can you give us a little insight as to what is going on in terms of the future for this CAR T?
Right, yeah. So there are several different trials with this CAR T product moving forward in the US. As you mentioned, this was mostly the phase one part of the study. We have the phase two part of the study for relapse refractory disease. We also have studies now that are for frontline newly diagnosed patients, both transplant eligible, transplant ineligible. Also for patients with earlier line therapy after 1 to 3 prior lines. So all of the different spaces pretty much are going to be covered with this product, hopefully.
So it seems like we have a lot happening in the era of CAR T cells. We certainly have two marketed products. There may be a third coming down the pike in a few months' time. And there is yet more targets that are being explored, GPRC5D. They're dual targeting like this. There are others that are targeting in GPRC5D with BCMA. So I understand that the portfolio of drugs in development for myeloma is still very robust.
Yeah, it's an exciting time with, not only the products that are already approved, but think top products like this that are in testing that really are going to challenge the ones that are already approved. And, between dual targeted CAR Ts and trispecific antibodies and combinations of bispecific antibodies, the future is very exciting for immunotherapy of myeloma.
Well, I want to thank you for taking the time to talk to us about this exciting research that you've been involved with, and hope I can draw on you for future appearances to tell us about how this and other projects that you're working on progress.
Definitely happy to help. Thank you so much for having me.
Thank you.